right, well, hopefully everybody is seeing my screen at this point. Yes?
Yes.
All right, good. All right, so thanks a lot, Gary, for the invitation to talk today. I really appreciate it. The name of this talk is They Have Tails, Incorporating Striped Bass Migration Ecology into Management. And I'm going to kind of start at where most talks probably end, especially given where I'm speaking today. This project really emerged out of hallway conversations between Bill Hoffman, Mike Adine, and myself over a number of years. And then, as you can see from this long list of people, and I'm probably forgetting several people here, it's a project that was only really possible because a ton of people at DMF participated and are continuing to participate in it. The amount of support we've gotten from multiple programs within the division is really amazing. Totally indebted to the agent growth folks for the ton of amount of work they've done. All of Bill's past and present FDI employees, my seasonals, Bill's temporary technicians, a lot of the folks down in New Bedford contributed to this either through helping with field work. A lot of the divers have helped us recover or put out receivers. So I really want to say one of the things I've always enjoyed about this project is that it touches a lot of different people within the division and a lot of the people's expertise that otherwise it just wouldn't be possible. So I think that's one of the great things, that it highlights what the division is capable of. And as you can see from these pictures, it's also a lot of smiles. It's been a lot of fun. It's been a good time, which makes it even better. A lot of time, but a really great time. So I'll launch into talking about striped bass. I think this is probably a fairly striped bass knowledgeable crowd, but here we are, the basics of striped bass. I love this figure, even though it's kind of a cheesy USGS figure, because it shows the introduced and native ranges of striped bass. And here you can see in the east coast of the United States, the northwest Atlantic. That's your native range. Going into the Maritimes, this is just a US map, so it doesn't really show too much into the Canadian Maritimes. But also all that red on the figure is where, as Caucasians spread across the United States in the 17 and 1800s, and, they decided to bring striped bass with them because they liked them that much. So it's a bass, it's a fish species that we really value, and we've ended up introducing all over the place. And obviously, as an anadromous fish, it does show some ability to landlock and become an entirely freshwater species. Also, within the more recent past, it's been at a fairly high level of abundance. This is from this 2018 benchmark stock assessment that Gary did so much work on. He probably never wants to see this figure again. Sorry, Gary. But while the spawning stock abundance has been coming down some in the last decade, it really hit a high peak for a sustained 20 years. That means that it always was a very important and popular recreational fish, and has reached new heights in the last few decades. Why not? It brings families together, makes an excellent trophy opportunity when you take the fish and throw it into the forefront of the camera. And it's also quite tasty if you choose to take it home. And Massachusetts is obviously a very important fish for Massachusetts. Gary wrote a great paper about this that came out in Northeastern Naturalist a few years ago. Here you can see on the left, the recreational value, so the percent contribution by weight of striped bass in Massachusetts. It's every year it's the most most important species when you look at weight, and it also likely contributes somewhere around $500 million or more of economic activity annually. Commercially, it's certainly not the most important species, but it's not inconsequential at all, with over $3 million of commercial landings in 2019, just in landings, and not talking about any of the other activity as far as fuel and bait and everything else that goes with that. And what's interesting is that Massachusetts is also very important to the coastal fisheries. So here you can see coastal estimates by state. These are the 2019 numbers of recreational effort, catch, and commercial landings. This is a fairly typical pattern where Massachusetts is usually between the first and third most important state as far as recreational effort and catch. And then commercial landings is all quota based. So every year we are the third highest level of commercial landings. So we're talking about fish species that is both very important to Massachusetts, and Massachusetts has a fairly outsized effect as far as the coastal abundance and the harvest of the fish. And as such, we've been fairly involved with striped bass research over the years. Before this study, the most recent was a study that was launched to look at how striped bass use the exclusive economic zone, the EEZ, and there was, in 2006 and 2007, there were a lot of fishermen were seeing large bass aggregations on Stellwagen Bank. So this was outside of the EEZ, outside of the 3-mile limit, where striped bass retention is or fishing is legal. Outside of that, you shouldn't be targeting striped bass and certainly not keeping them. It's illegal. Back in the 80s, they made that a sanctuary zone when the abundance was very low. So all these fish were out on Stellwagen, a lot of people fishing out of Stellwagen, catching striped bass, and basically saying, hey, well, this isn't good. All these fish are inaccessible to us, whether it's recreational or commercial, and they're sitting out here and we can't get at them. So a lot of fishermen in Massachusetts wanted to be able to get at these fish, wanted to know if they were kind of just staying out there and never being able to be caught inshore. So as a result, DMF conducted an acoustic study, and I'll show some of that results for other parts of it, but they wanted to look at the transboundary movements and see if EEZ was indeed a refuge, and what they found was that it was not. So this was good. It showed they used acoustic technology to show that fish were coming in and out of state waters over the course of the summer. They did it every year, and they were indeed, individual fish were indeed accessible to our fisheries. As part of that work, Bill and Micah were also able to examine migration routes and stock composition of fish in Massachusetts. And really, these were all Stellwagen Bank fish that they tagged. And what they found was that in the spring, these denoted by these green arrows, fish would largely use the Cape Cod Canal to enter Massachusetts waters, with a smaller proportion of fish coming around the backside of the Cape. But in the fall, most fish would exit around the backside of the Cape, with a smaller proportion going through the Cape Cod Canal. So that said something about where fish were available and when they were available to recreational and commercial fisheries. Utilizing other people's arrays that are put out along the coastline, they were also able to take a look at where our fish were coming from, which natal populations were contributing to Massachusetts fisheries. And they found a mix, Chesapeake Bay dominant mix. It is different depending on which year they looked at, but Chesapeake was the dominant in both. This contrasted to some previous results, which again showed variability, but Berggren and Lieberman in the 1978 paper, where they used morphometric analyses, said that all fish caught in Massachusetts waters were from the Chesapeake. And then Mary Fabrizio in 1987 used morphometrics as well as eye-lens proteins. And she showed a mix in one year quite dominated by Chesapeake, and then another year much more by Hudson fish in the samples she received. And I will point out that Micah and Bill brought in Jeff Kneebone, who wrote up both of these papers and did a really great job. And so if anybody is interested in more detail on those papers, they are out there and part of our contributed series that Mike administers. And of course, Mike was the mad scientist behind all of this. So one of the big reasons that people look at this and are concerned about it is that there's a great deal of management ambiguity when it comes to striped bass. So here you can see the three major contributors as far as spawning populations. They're anadromous fish. So in the springtime, they're running up into fresh water or estuaries to reproduce. And then eventually these the young of your fish grow, maybe 1 year, maybe several years, depending where they are and which fish they are, and then come out and join what is called the coastwide population. And because we don't have a good way to identify these fish back to their individual populations, this is just how we do it. We say there is a coastwide population. So basically we're taking all these striped bass from all these different runs and making them into one big striper pie that we then divide up among states. And so that's one issue that's been very long standing with the way that we manage striped bass that I think a lot of people would like to see change. And about the time that Bill was wrapping up that last project and I came to DMF and we started talking about all this stuff, there was also a change in our commercial fishery that a few of us were a little bit concerned about. And it was that during the early 2010s, especially late 20aughts, there was a transition in where fish were being landed, commercial fish were being landed in the state, with a shift out of all the other counties going down, and Cape Cod, Barnstable County, basically accounting for more and more of our commercial fishery. You did not need a bar graph or landings to know this. It was a really popular topic in the media. There was kind of like a warfare going out on commercial fishing warfare on the days when a lot of anglers having issues, a lot of parking lot issues, a lot of towns on the Cape not enjoying exactly how many anglers were coming down. And it did lead to a lot of adjustments to management, where we now have fewer open days, and we're trying to get our effort out over a larger part of the summer, and which has had benefits, I think, for the fishermen as well, as the price has been better, a little bit less of a derby fishery. But we were concerned about what this might mean, because what Bill's data showed was that fish were kind of had predictable areas they were going to, had predictable ways in and out. And if you had just these one group, one or two aggregations that were getting hammered on by high levels of fishing, then what would that mean both for Massachusetts fish and for coastwide fish? So knowing all that, we embarked on a new project with a couple big picture goals. And the first was to examine the year-to-year stability of mass striped bass aggregations. And we also wanted, as I just hinted to, determine potential differences in mortality of aggregations with a number of different factors that could influence that. And finally, kind of had this long-standing goal of improving stock composition, identification, and monitoring. And in the big picture, what this is really about is, over the last 20 years, the migration ecology of fishes is a subject that's, as we've gotten more and more improved techniques to look at it, I think everybody who's paying attention is very amazed by what these fish are capable of. All fish are capable of when they migrate, whether they're tunas or eels or striped bass or salt sea run brook trout. There's some really amazing patterns going on. It's happening in river fishes as people look now as well. But the struggle has been, how do researchers and managers take all this increased understanding and wrap it into management and actually improve what we're doing? So in the big picture, each of these sub goals is about taking migration ecology and applying the lessons we've learned to improve the management of our resource. So the way we aim to do that was by combining acoustic telemetry and genetic stock identification. And we broke the state out into 3 study areas, one in southern Mass, Buzzards Bay and Vineyard Sound, a second off of the backside of Cape Cod, and then a third in Boston Harbor. And so I'm going to talk for a little bit now about the acoustic telemetry portion of this study. And we did tag bass in these 3 areas. You can see that in over 2 years, you can see the numbers in 2015 and 2016 in all 3 of those areas. So we tagged more than 75 fish, aiming for at least 25 fish in 3 size categories, because we're interested in how size could affect mortality. We did surgical implantations with the largest tag that Vemco makes, and at that point they were getting over 7 years of battery life the way we had them configured. I think that's over 10 years now. So you can really get some great longitudinal information about individual fish. The other half of that, of course, is putting out all those receivers in coastal waters. And so in the final 2 years of our study in 2018 and 2019, this is pretty much what our Massachusetts array looked like. So you can guess that that's whenever you see us all going out on boats to do this stuff, that's what was going on. Or if you're in Gloucester out in the parking lot, you can see us staging everything and cutting rope and doing all that and splicing ropes. So in this figure, all of the blue dots are receivers managed by Greg Skomel or somebody else in New Bedford doing research, doing acoustic research, and the red dots are out of Gloucester, and the red dots are primarily the striped bass with some habitat environmental monitoring as well. But you can see it's quite an array, well over 100, 150 receivers every year. And we used a bunch of different ways to do this, but you can see in the bottom right, you have some things that go on aids to navigation, and then you have setups to put down on the bottom. And this is part of these bags are part of our pop-up setups, because we leave those at the bottom so those can decompose and not contribute any waste to the ocean floor. Outside of Massachusetts, this is also a great time to do this study, because there was very robust coverage in all these spawning rivers for striped bass on the seaboard. And what you can see here, each little dot here, and they're color coded, you can see the legend, represents a receiver that one of our striped bass at least was detected on. And at last count, we were up to 701 different receivers that where a striped bass have been detected. So it's pretty amazing. And as I said, the nice part is we had robust coverage in the Hudson River, the Delaware River, and then in Chesapeake Bay, as well as this gate off of the Chesapeake. So we knew if fish were going in, at least, even if we didn't get them in a river. And what that really meant was that we were able to make this jump for striped bass that other people have done for different species, where we're going from this kind of analogs. This is what you would have as your diagram for striped bass migration. Now we can go into the digital age and you get something like this. And so these are all the striped bass that we tagged in 2015 and 2016, moving in coastal waters. And you can, along these receivers, it's going by a weekly time step, and the bigger the dot, the more fish were detected at the receiver that week. So now we're getting into the fall. Fish are coming out of Massachusetts, going down the shore, off Delmarva, off of Cape Henry in Virginia, come around back into the spring, and you're going to see the fish start to go up the Chesapeake, into the Delaware, the Hudson, through May. And by the time you get into May and June, now the fish are back in Massachusetts for the summer. So this is really a big leap. Now we have this actual illustration of how fish are using the shoreline, and we can track them really, down to a daily time step if we have enough receiver coverage. So you just saw some of that spawning count activity on a big scale. Let's look at it a little closer in. This is called a chord diagram. For those of you who've never seen one before, it's not the most often used way to display data, but I like it a lot. So each tiny little bit of one of these ribbons, and hopefully everybody can see my mouse, represents one fish. And on the top half of the chord diagram is where we tag the fish. So you have Buzzards Bay, Cape Cod, and Boston Harbor. And on the bottom half is where fish ended up going to spawn the following spring. So hopefully that makes sense. You're connecting places we tagged to places they went to spawn. And as you can see in the text bullet here, we actually had a fairly low number of fish that we detected in spawning areas. We suspect that that's a size effect. I haven't gone and looked into it too deeply yet. But a majority of those fish, nearly 70%, were located in the Hudson, and all three of our aggregation areas had a similar composition of fish. In 2017, we had more bass available, but even fewer fish were actually detected in spawning estuaries during spring. And now you can also see that there's more fish that were connected to the Delaware system. We did indeed have 23 repeat spawners, 3 of which actually, most interestingly, were detected in the Hudson one year and the Delaware the next, or vice versa, or maybe the Chesapeake. Which for striped bass, although we don't really know this, we assume that they have a very high natal fidelity. So it was interesting to see that there was some shifting of natal areas during the spring, or spawning areas. Here's Frank's Zappa to remind us all that it's not the mean that's important, it's what's going on around it. Thank you, Frank. And again, majority of the fish in the Hudson. For this year, this time there was a difference in the aggregation composition. As you can see, Cape Cod, unlike Boston Harbor and Buzzards Bay, had fewer individuals being connected to the Delaware. Now comparing the 2 years, 2016 and 2017, you can see that they are different. Again, that Delaware effect. I've also started to look at a size effect for these years. So here it is broken out by size. And as you can obviously see, for both the sublegal fish and recreational fish, it was very heavily Hudson-based fish, whereas for the bigger fish, there were a lot more Chesapeake fish. Slightly different in 2017, but in both years, there was a difference based on the size of the fish. So moving from spawning areas to aggregation fidelity, which was, we were all very excited to look at this because we had a feeling that what we would see based on past work and just looking at individual histories, but it's turned out to be pretty interesting, especially given the results of the last study. So as a reminder, all the receivers are color-coded here. based on the where fish were. I've introduced Cape Ann North, I'll get into that in a second, as a fourth or fifth area. And the canal just to have it separately, but you have Cape Ann, Mass Bay, Canal, Cape Cod, and Buzzards Bay Vineyard Sound moving from north to south. All the receivers are colored. We tagged in discrete areas, but because of our receiver coverage, we're able to talk about larger areas. So from on here on in, when I talk about our results, I'm talking about fish detected over pretty broad areas. Here's another chord diagram. I'm going to walk you through this one as well. On the top here, you'll see year 2, and on the bottom, Y3 is year 3. And since we made every effort when tagging, we made it hard on ourselves by not tagging in June, which is typically the best month for striped bass, among the best months, or October. So we tried to keep our tagging to the dog days of summer, thinking that that's when striped bass would be locked into their summer area. We did have some fish, especially in Boston Harbor, that we tagged, that ended up spending most of their summer up closer to the Merrimack. So we wanted to capture that. I may end up bringing those back into just a larger northern Mass area. I think it's somewhat of an artificial designation on my part, but for here it is. So we did say that the area they spent their second year in, where they were free of tag effects, is what they really wanted to do, where they were actually going right where they wanted to be. So year 2 is on the north, year 3 is on the bottom. So for 2015 tagged fish, that's 2016. For 2016 tagged fish is 2017 for year 2, and add 1 to that for year 3. The long story short is that what you see is fairly high to remarkably high aggregation fidelity year to year for these fish, especially for Cape Cod fish. Cape Cod seems to be more of a donor area, or sorry, a donee area than a donor area. Other fish will transition to using Cape Cod, but fish that we tagged in Cape Cod really had a very, very high fidelity to Cape Cod waters. Another way to look at that is to talk about fish that we have longer time series for. So for a fish we tagged in 2015 and we had records of through 2018, we saw a great number of those over all 4 years remain in the same area. So of the 54 fish, we tagged 175 fish in 2015. We had 54 striped bass that survived enough of the summer of 2018 for me to make a designation. 90% of those fish had remained in the same area for all 4 years that they were at large. So that's pretty remarkable. And I also want to use this opportunity to transition to talking about mortality a little bit. So as I just said, we tagged 175 fish in 2015. By 2016, that was 134, 85 in 2017, then 54 in 2018. So you're seeing a yearly reduction in the amount of fish available to us. And those overall annual losses were pretty consistent. As you can see here, you have the by tagging aggregation returns to Massachusetts water. There's an asterisk on annual mortality here, in that we're assuming that we're going to have 100% coverage in Massachusetts waters, and that we're not going to have tag loss. I almost don't think about it. Micah was nice enough to point that out. I should put an asterisk on it, because with the surgery method we use and the amount of receivers in Massachusetts waters, I really think that those are some pretty safe assumptions. But you can see about a 30% annual loss over the course of the study. And this is going to be the next big step for me in analyzing these data sets, is to get into all the other factors that could go into that and try and make a model that incorporates them. But I do want to briefly talk about one thing that might be affecting that, and that's migration corridors. So here is a figure. Again, everything is color coded by where fish spend their summers, their aggregation. And on the bottom here, before the dash, the words before the dash show the migration route into Massachusetts, and then the after the dash for each bar is the way they went out. So these are fish that went into Buzzards Bay and left by Buzzards Bay. They never went anywhere else in Massachusetts. Fish that came into Massachusetts waters through Buzzards Bay and then the canal, likewise then back out through the canal. Canal Cape, Cape Canal, Cape Cape. And what you can see here is that fish that exit the canal north when they come in the spring, so fish that are going into Massachusetts Bay, that are going onto the North Shore, almost entirely leave through the canal. Only one of those fish went around the backside of Cape Cod. And so that's completely different than what Bill and Micah saw in that first study where they just tagged fish on stellwagen bass and we started making assumptions about how all Massachusetts bass work. What we can see here is that fish that are coming into the northern part of the state are doing something different and really favoring using Cape Cod Canal to both enter and exit Mass waters. This was a pattern that was a little bit more complex in 2018, but repeated. It is also pretty interesting. You can see there's some more categories here. And these ones that say Buzzards Bay, Vineyard Sound to something else, what happened here is that for the first time we saw fish going through Woods Hole or one of the other holes in between the Elizabeth Islands and then coming around Cape Cod. And I was pretty puzzled by this until I started talking to some charter fishermen and who typically rely on that springtime canal run for their early season charters. And they said that the canal was dead in spring of 2018. There was a huge group of bait, both squid and butterfish in Nantucket Sound that year, and that's where the fish were. So it was interesting that we also saw that in our acoustic telemetry returns. But the big story again is that there's only one or two fish, or one fish that's going from the northern part of the state that's going to exit by Cape Cod. So again, these fish are going in and out of the canal for the most part. How does that link to mortality? Well, in 2017, as an example, 30% of the bass that entered Massachusetts waters did not leave. And that's fairly typical. We see a lot of our mortality of our bass within Massachusetts waters. And we can look at where the last detection of a fish was as a proxy for where it died. And what you see is that, oh, sorry about that, I apologize. What you can see is that there's quite a bunch of fish that had their last detection in the Cape Cod Canal area. This is color-coded by spring, summer, and fall for green, red, and orange. Especially that fall run, that kind of epic and much hyped fall canal run was the majority, or was the highest level of mortality for our fish. And it turns out that, as you could guess from the migration patterns, those were all fish that summered in the northern half of the state. So just about half of the total mortality that's happening is on one aggregation in that year, rather than spread evenly among the three. I'm going to shift now and talk a little bit about the genetic work we're doing. This has been a really interesting partnership between Mass DMF, UMass Amherst, now University of Montana, as the Amherst professor who was started there is at Montana now, and then Dr. Scott Pavey at the University of New Brunswick, St. John. And so we have quite a number of samples. We're using single nucleotide polymorphisms, or SNPs, which are generally the state-of-the-art marker now for doing these types of examinations. And in our collaboration with Dr. Pavey, it's also going to allow us to look at some really interesting adaptive behaviors and how they could be linked to the genome of these fish. We have completed the baseline results and published them. You saw in that last figure the 15 different rivers that we have fish from. And this is going to be important for the mixed stock analyses I'm going to be doing over the next few years with all those commercial and recreational samples. But basically, there are 6 separate spawning groups that we could identify from the SNP markers. The Canadian groups are really well differentiated, whereas the US groups were far closer genetic-wise related to each other and harder to discern from each other. But for the US rivers, it turned into 3 different groupings, one for the Hudson, another that for all of the Chesapeake Bay runs and the Delaware River, and then a third for the spawning populations in North Carolina. However, the good news there is that it seems like we're going to be able to do really high accuracy classifications, especially for the mixed stock estimates. Maybe not individual assignments, but mixed stock estimates are going to be great. Here, using the very large number marker panel they had for that first paper, they just showed that over 99% assignment accuracy back to their population of origin, which is a really good initial look at how we're going to be able to do as far as mixed stock estimates. We're trying to get money to finish off all this work. Mike and others did a great job of helping us get recreational license money in 2018 and 2019 to get this process started. And I'm hopeful that in the next month or two, I'm going to hear back about an ACCSP grant that not only would give us enough money to finish the genetic samples, because I said we had about 5,000 of those in the last slide. It would also help us to develop a program where that would be put into data streams and hopefully become an annual monitoring aspect up and down the coast for each state. It should be relatively low cost and low effort, not too much different than everything that's going on now when samples are being collected for ages. So it would be a really big leap forward in what we would be able to know about the species, and then what people like Gary would be able to do with the models if we can ever talk him into doing any more striped bass work. We could finally get to this kind of population-based management that everybody's been chasing for quite a while. So for now, my conclusions, we've made some good headway into the stock composition and stability questions. We really need the genetics at this point for this question. It's going to boost our sample size tremendously and let us see a lot more about what's going on. And then our hope is that the fish we tagged are representative of what's actually being caught in each area, and we can apply some of these behaviors we're seeing to larger areas and pools of fish. We showed a pretty remarkably high fidelity to summering area, and I'm looking forward to honing in on that and looking at tagging date and sizes and other things that might be affecting it. And that's part of my general path forward at this point. There's kind of a tag lag that goes on in every acoustic telemetry project where we always have a fairly good idea of what's going on in our arrays, so in Massachusetts waters, but I'm always waiting for detections to filter in a year and a half later from other groups up and down the coast. So now in January of 2021, I'm fairly sure I have all my 2019 data. The study, as far as we're concerned, is going to go through 2019, so I can start really getting into the entire data set, and also pushing forward with genetics and mortality components, and hopefully coming up with a model talking about how all these different factors could possibly lead to differential mortality for striped bass in Massachusetts. So that was a lot. Thank you everybody for sticking with me. I saw a few chat things pop up. I don't know what they said, but I'm really more than happy to take any questions now.